Hello everyone, welcome to today's session on biosynthesis of cholesterol. I am the facilitator Dr. Mausmi Shankar Adala, working as an assistant professor in the Department of Biochemistry, St. Joseph's College for Women, Autonomous. In today's session, the contents of cholesterol biosynthesis pathway were explained under different categories, namely mevalonate formation, isoprene units activation, squalene formation, and finally cholesterol formation. This is much more precisely and in brief detailed description was given under acetyl-CoA is converted to mevalonate, how mevalonate is phosphorylated and decarboxylated. Also, the isoprene units from squalene formation, and finally how squalene is converted to cholesterol. As we all know, cholesterol is a 27 carbon compound and it is formed from the basic fundamental unit, that is a starting compound is the acetyl-CoA. Before, cholesterol is formed as 30 carbon molecule. Later on, by undergoing certain processing, it is finalized to 27 carbon molecules. In day-to-day life, people usually get scared listening to cholesterol, feeling that it is a bad fat or it is not required by the body, and excess of that leads to any coronary problem, that is heart problems. But in general, minimum based on your body weight, there is a particular percentage prescribed by the WHO, where based on that your body should have minimum amount of cholesterol that is must and should useful for your body metabolism, that is your lipid metabolism. So keeping in that, cholesterol need not always be harmful. The minimum amount of cholesterol that is required by the body has to be supplied either through your diet, because usually once you eat your food, excess of carbohydrates is also converted into cholesterol. How? The excess of carbohydrates is first converted into acetyl-CoA, and this acetyl-CoA, being the parental compound or the starting molecule of cholesterol, helps in the formation of cholesterol. Going into the biosynthesis procedures, we have 4 different stages as we have mentioned in the contents. The first stage is formation of mevalonate from acetyl-CoA. Here, the first step is condensation of acetyl-CoA molecules. 2 molecules of acetyl-CoA, which are 2 carbon each, condenses to form aceto-acetyl-CoA to form 4 carbon compound. So, the aceto-acetyl-CoA is a 4 carbon compound. So, this reaction occurs only in the presence of an enzyme called as thiolase. So, under the reaction format, we very clearly mentioned acetyl-CoA plus acetyl-CoA under the influence of the enzyme thiolase results in the formation of aceto-acetyl-CoA along with coenzyme A, which is having a sulfhydryl bridge. The second step in the cholesterol biosynthesis is formation of HMG-CoA. HMG stands for hydroxy 3-methyl-glutaryl-CoA. Here what happens is aceto-acetyl-CoA, which is formed in the first step, condenses with another acetyl-CoA to form 3-hydroxy 3-methyl-glutaryl-CoA. So it is abbreviated as HMG-CoA, and this HMG-CoA is a 6 carbon compound. It is a 6 carbon compound because aceto-acetyl-CoA is 4 carbon. It condenses with acetyl-CoA, which is 2 carbon. So 4 plus 2 gives 6 carbon. Henceforth, 3-hydroxy 3-methyl-glutaryl-CoA is a 6 carbon compound. In this reaction, the major enzyme involved is HMG-CoA synthase. I repeat, HMG-CoA synthase. As a reaction part, you can see aceto-acetyl-CoA, which is 4 carbon, combines with acetyl-CoA, and here one molecule of water is also involved. In the presence of the enzyme, HMG-CoA synthase is converted into HMG-CoA plus coenzyme A with sulfhydryl bridges. The next step is a reduction of HMG-CoA to mevalonate. HMG-CoA, which is a 6 carbon, is reduced to mevalonate. That is, there is no change in the carbons, only there is a change in the structure. Mevalonate using NADPH as an reducing agent. We all pretty well know NADPH and FADPH are called as reducing equivalents, and these are involved in the ATP synthesis. These enter the ETC to produce ATPs, and here the enzyme involved is HMG-CoA reductase. This is the rate limiting step, that is whether the cholesterol biosynthesis has to move forward or it has to take a halt, depends upon this enzyme availability and the action of this enzyme. Now what is the site of action of this particular step, since it is a rate limiting step? This occurs in the smooth endoplasmic reticulum, which is abbreviated as SER. So this being the rate limiting step, it is also called as regulation point, that is HMG-CoA reductase, that is the enzyme used for conversion of HMG-CoA to mevalonate, is highly regulated. How? Through a feedback inhibition by cholesterol or the hormonal control by insulin or glucagon and phosphorylation. That means whenever there is enough cholesterol produced already inside your body, when there is no more space for the storage of this cholesterol, immediately this enzyme will be blocked. That is called as feedback inhibition. Secondly, insulin and glucagon. What is the function of insulin? To regulate the glucose concentration. So here what happens is, when there is excess of glucose, there will be formation of cholesterol. But when the cholesterol itself is not having any more place or storage capacity in the body, so it is showing feedback inhibition of this particular enzyme. Henceforth, insulin also will show its own control, even glucagon and phosphorylation also shows. So this can be explained in the form of the equation HMG-CoA plus 2 molecules of reducing equivalents, that is NADPH, and 2 protons are also involved, and this comes up to mevalonate plus 2 NADP plus because 2 hydrogens are already used for the reduction of HMG-CoA and to form the mevalonate, and finally again as usual coenzyme A with sulfhydryl bridges. The enzyme, as we have mentioned, HMG-CoA reductase, is showing its action. Coming to the stage 2, which is a conversion of mevalonate to activated isoprene units. Phosphorylation of mevalonate, and mevalonate is phosphorylated. Phosphorylated is nothing but addition of a phosphate group. And from where we get these phosphate groups to add? Naturally from the ATP. So phosphorylation of mevalonate step involves the addition of a phosphate group to mevalonate, and this results in the formation of 5-phosphomevalonate. I repeat, phosphate group is added to the mevalonate to form 5-phosphomevalonate, and this occurs in the presence of the enzyme mevalonate kinase. So mevalonate plus ATP gives 5-phosphomevalonate plus ADP. Why ADP? Because since what is the full form of ATP? Adenosine triphosphate. So here we are having 3 phosphate groups. Here 1 phosphate group is added to mevalonate to form 5-phosphomevalonate. Thus there are only 2 more phosphates available. So we have written here ADP, and the enzyme involved here is mevalonate kinase. Kinases are the enzymes that are useful for addition of phosphate groups. On the other hand, phosphatases are the enzymes that removes the phosphates from the group. So hence mevalonate kinase is nothing but this is the enzyme that adds phosphate onto mevalonate, taking the phosphate away from the ATP and forming ADP plus 5-phosphomevalonate. Further phosphorylation is seen, that is 5-phosphomevalonate is further phosphorylated again to form 5-pyrophosphomevalonate. 5-pyrophosphomevalonate. And the enzyme here involved is phosphomevalonate kinase. Usually the names of the enzymes take up their name from the substrate on which they are adding. Here very clearly we can see phosphomevalonate kinase is nothing but kinase adds the enzyme On whom it is adding the enzyme? What is the substrate? Phosphomevalonate, 5-phosphomevalonate. Okay, so thus the enzyme phosphomevalonate kinase adds ATP. From where it takes the ATP? I mean phosphate group. From where it takes the phosphate group? From the ATP molecule, and this results in the formation of 5-pyrophosphomevalonate plus ADP, and this results in the formation of further phosphorylated compound, that is pyrophosphomevalonate. The next step is decarboxylation to isopentenyl pyrophosphate, and from here there will be some abbreviated forms, that is isopentenyl pyrophosphate is nothing but IPP. I repeat, isopentenyl pyrophosphate is IPP. 5-pyrophosphomevalonate undergoes decarboxylation to form isopentenyl pyrophosphate, that is IPP, a 5-carbon activated isoprene unit. Here the enzyme involved is mevalonate 5-pyrophosphate decarboxylase. So whatever compound that is formed in the previous step, that is 5-pyrophosphomevalonate, we are removing carbon dioxide. Naturally the enzyme that removes the carbon dioxide is nothing but decarboxylase enzyme. So definitely when the carbon dioxide is removed, we get IPP. IPP is nothing but isopentenyl pyrophosphate. Along with this, here also for this reaction ATP is required, and this ATP phosphate group is involved, the energy is involved, and it results in the formation of ADP plus carbon dioxide. Why carbon dioxide? Because the carbon dioxide is removed from the substrate. Seventh one, isomerization to dimethyl allyl pyrophosphate. It is abbreviated as DMAPP. IPP isopentenyl pyrophosphate is isomerized to dimethyl allyl pyrophosphate, that is DMAPP. DMAPP. The enzyme involved here is isopentenyl pyrophosphate isomerase. Why isomerase? Is because of even IPP is 5-carbon compound, it is again converted into dimethyl allyl pyrophosphate, that is DMAPP, that is also 5-carbon compound. Hence one compound, as the isomers we know, they have same formula but structural difference. Similarly here the IPP is converted into DMAPP, thus the isopentenyl pyrophosphate isomerase converts this. So now we are having DMAPP. Stage 3, that is the formation of squalene, condensation to form geranyl pyrophosphate. Now the IPP which is formed in the 6th step and the DMAPP which is formed in the 7th step, both are 5-carbon compounds each. So they both condense to form geranyl pyrophosphate. I repeat, geranyl pyrophosphate GPP, that is a 10-carbon compound. Here the enzyme is geranyl transferase. So this results in the formation of geranyl pyrophosphate GPP plus pyrophosphate. Further condensation of farnesyl pyrophosphate, that is FPP. Now the GPP that is formed in the 8th step, that is the previous step, is of 10-carbon compounds, and it reacts with one more IPP, isopentenyl pyrophosphate, which is 5-carbon compound, to form FPP, farnesyl pyrophosphate. I repeat, farnesyl pyrophosphate FPP, which is 15-carbon compound, and the enzyme involved here is farnesyl transferase. Farnesyl transferase. We can see very clearly in the reactions, GPP plus IPP gives FPP, that is farnesyl pyrophosphate, which is 15-carbon compound, along with the pyrophosphate is formed. Formation of squalene, 2 FPP molecules, that is 15 carbon each, combine to form squalene, that is 30-carbon compound. I repeat, 30-carbon. The enzyme involved here is squalene synthase, and this step requires NADPH for reduction. In the reaction it is very clearly shown, 2 FPP molecules, that is 15 carbon each. So 15 into 2, 30 carbon naturally. So they undergo a reduction procedure, and this results in the formation of squalene along with 2 pyrophosphates and NADP+. Stage 4, that is the last stage, conversion of squalene to cholesterol. Epioxidation of squalene, that is squalene is converted to squalene 2,3 epoxide by the addition of an oxygen atom, and the enzyme involved here is squalene monooxygenase, and this uses oxygen and NADPH. So squalene plus oxygen plus NADPH plus H plus gives squalene 2,3 epoxide plus NAD plus water, and the enzyme involved here is monooxygenase, squalene monooxygenase. Coming to the last but one step, cyclization of formation of lanosterol. Squalene 2,3 epoxide undergoes cyclization to form lanosterol, the first sterol structure with 4 rings. The enzyme involved here is oxidosqualene cyclase. So here the squalene 2,3 epoxide is converted into lanosterol, and this results in the presence of the enzyme oxidosqualene cyclase. I again repeat, oxidosqualene cyclase. And the last step, conversion of lanosterol to cholesterol. Lanosterol undergoes 19 steps involving. These include the main things like demethylation, reduction, isomerization. These steps remove 3 methyl groups and rearrange the double bonds. The key enzymes involved are lanosterol, 14-alpha demethylase, alpha-24 reductase, etc. So the lanosterol, that is the 19 steps demethylation, reduction, isomerization, cholesterol is formed. The summary table, here the process is acetyl-CoA is converted to mevalonate, the key intermediate is mevalonate, and the key enzyme is HMG-CoA reductase. Second one is mevalonate is converted to isoprene units. Here the isoprene key intermediates are IPP and DMAPP. Here the enzyme is mevalonate kinase, and the third one is isoprene units, which is converted to squalene. Here the squalene is a key intermediate and the enzyme involved is squalene synthase, and the fourth one is squalene is converted to cholesterol. Here the lanosterol is converted to cholesterol, it is a key intermediate, and the enzymes involved are squalene monooxygenase and lanosterol demethylase. Concluding today's session, cholesterol biosynthesis is a complex multi-step process essential for the production of cholesterol, a vital lipid molecule critical for the cell membrane structure, hormone synthesis, bile acid formation, and vitamin D production. The pathway begins with the condensation of acetyl-CoA units to form mevalonate, proceeds through several phosphorylation and decarboxylation reactions to produce activated isoprene units and culminates in the synthesis of squalene. Squalene then undergoes cyclization and a series of modifications to form cholesterol. The key regulatory steps in the reduction of HMG-CoA to mevalonate by the enzyme HMG-CoA reduction, which is tightly controlled by feedback inhibition by cholesterol itself. Cholesterol biosynthesis is energy intensive, requiring ATP and NADPH, and occurs primarily in the liver, as well as in other tissues like the intestines and adrenal glands. Thus, the biosynthetic pathway not only ensures a continuous supply of cholesterol for physiological needs, but also integrates with broader metabolic and hormonal controls to maintain lipid homeostasis in the body. Thank you one and all for your cooperation and support.